Yeah, so Gordon is an associate professor in the Department of Biology at Emory. And before coming to Emory, he did his postdoc with Bill Biolek and Joshua Chavez at Princeton. And before that, he did his PhD at Cornell in physics. So Berman Lab is interested in solving various problems using tools from theoretical physics. And then especially they're interested in animal behavior and other interesting problems in biology. So with that,
Thanks, Hannah. So, thank you for everyone coming out today. It's a really full room. This is great. And especially thank you for having me speak at a neuroscience seminar, which I'm going to show you very little neuroscience. So we're going to talk about the end outputs, thinking about what behaviors, or the end output of what the nervous system is doing. And what a lot of my group does is sort of the opposite of what a lot of sort of biological science does, where you measure tiny little things and try and go up to the big thing. We're going to the big thing and try and work our way back towards the middle. And hopefully these two approaches kind of meet together and say interesting things. So I'm going to talk about kind of my group thinking about behavior. And particularly, I'm going to focus on this notion about variability or variations in behavior. One of the reasons why I have all this music stuff on the front page is one of the things I like thinking about in behavior is what's the right metaphor? What's the right way to think about the structure? And what's commonly done in almost all sort of things when people are thinking about what's the structure of behavior, the metaphor people like to use is words, linguistics. And what my kind of way of thinking about things tends to be is more of this musical way of thinking. And hopefully I'm not going to explicitly talk about like direct musical connections to that idea today. That's actually a whole other weird, crazy subproject. But really thinking about this more sort of continuous amorphous structure, which allows us to bridge scales from stuff going from like fast muscle twitches all the way up to aging, thinking about how to do that. And really the notion about thinking about is this notion about variability. And the problem about variability is this is sort of what we really are dealing with most of the time when talking about variability of behavior, with sort of apologies to Raymond Carver, Edward Hopper, and Banksy all at the same time. What we talk about when we talk about variability in behavior, because animal behavior is very strange. And oftentimes what we sort of ascribe to biological messiness is really the interesting variability going from what one animal is doing or what different animals are doing, what animal does from time to time to time, and what that animal is doing between different contexts. And how do you sort of make sense of this? Can we build mathematical formalisms to try and just actually put numbers on this really complicated field that people just often will write off as noise? That's sort of the goal. I'm going to show you the sort of today's talk is going to be kind of a through line through a lot of stuff that my group has done over the past few years, starting with some published stuff and then ending with some completely half-baked unpublished things that we're not even done with yet, but I just felt like showing. So we're going to start with that and let's go. And also please feel to interrupt me as I go along. I have plenty of slides I can skip. I'd rather just have a conversation. So again, sort of as Hannah mentioned, my group focuses on animal behavior. And in particular, we're interested in what I call either natural or naturalistic. Well, I shouldn't say I call, many people call it natural or naturalistic behavior. And here what we basically mean is things that are kind of outside of the realm of like a laboratory task oftentimes. So either animals in the wild or animals in somewhat, let's say, impoverished lab conditions, but they're still kind of given agency to do what they're wanting to do within that condition without having to like have some sort of operant conditioning or anything like that. And so we're interested in what are these types of structures. And one of the big challenges, how do I like put numbers on any of these things? How do I quantify what's going on this animal or the swarm or the school of fish or even this fly running around in the dish? So there's the part of this that I'm not going to talk about, which is one thing we've gotten really good at over the past, particularly like, let's say 5 to 10 years, is tracking the poses and the postures of animals. Things like deep lab cut, sleep, dance, everybody's favorite types of methods for going and tracking pause or positions or whatnot. We can do this. It still takes a lot of work, but we've gotten really, really good at these things. What I mean in trying to make sense of that is really trying to make sense of what are these longer timescale structures in behavior. Things like circadian rhythm or hunger or social state, parenting state or aging. And this gets into some fuzzy mathematical questions or even just experimental questions. The example I like to give is hunger. You can't measure hunger, right? You can measure kind of reflections on Plato's wall of hunger. You can measure what is an animal willing to do to eat? How much food did an animal eat? Like all sorts of things like that. But that internal state of hunger from the behavior alone, you can't measure. Maybe there's some physiological correlate that you can go after, but really that state of hunger is really something which is affecting a bunch of different behaviors that an animal is doing, and you kind of have to make an inference about what that's doing. And not only that, these internal states are all kind of yelling and fighting with each other. Another example I like to give is if you're laying in bed at night and are hungry, you decide to get up and eat. Right? So you have two internal states which are fighting with each other, circadian rhythm and hunger, and then which one of those wins is a result of a complicated interaction of internal states, and then which one of those wins then has longer timescale consequences that sort of reverberate on throughout the rest of your decide to get up and eat, you sort of have to live with the fact that you're now going to be a little bit more tired. And of course, so I'm saying all these things, and one of the things I really want to say, I'm not the first person to say any of these things, right? This is all couched in the literature from really from the 1950s to the 70s. There's this really deep literature in ethology, particularly from people like Tinbergen, Walter Heiligenberg, and yes, Richard Dawkins. This is the Richard Dawkins, well, who was actually a student of Tinbergen's before he became Richard Dawkins. He was actually just a mild fly grooming expert. Maybe not mild, but at least more subdued. So the idea is, particularly this book here, I've sort of probably browsed through a gazillion times, is Tinbergen's A Study of Instinct. And one of the things that really, what he proposed is this hierarchical set of internal states, thinking about how these internal states are interacting with each other, how they can be repurposed across different sets of behaviors. Then ideas from people like Walter Heiligenberg, we're talking about really what you then can do. In this particular case, he's studying one of Patrick's favorite organisms, cichlid fish, thinking about how do you actually understand how that repertoire of behaviors. So here, this is just painstakingly annotating a bunch of different behaviors like feeding or attacking or courting across time, and just seeing how the linear combinations of these behaviors are changing across time, and can we actually isolate what those long timescale structures are. And then Dawkins, in this really wonderful 1976 paper, was thinking about software principles for behavior, right? How do you get beyond? So he kind of derides what he calls the neurophysiologist nirvana of we're able to measure every single possible neuron in the brain, every single neurotransmitter. What now? And if we were to do this across multiple animals, what general principles would we come across? And so he makes this argument for really understanding the software structure. And that's kind of the spirit in which we're going to be approaching this, is can we have some sense of what that software might be? We're not going to get there today, but I just want to kind of at least show you kind of how we've been thinking about it. All right. So now, all right, broad intro over now, so let's get a little more specific. So this gets to this problem. The first thing we need to do is to be able to actually measure variability and define what that means. So usually in most neuroscience experiments, these are a couple great videos from some friends of the lab. This is from Megan Carey's group, watching a mouse locomote, or this is from Abby Pearson's group, watching a mouse reach for a food pellet. You have this kind of trial structure you can go by. Even in the somewhat more naturalistic behavior, right? You have an individual reach, you have a gait cycle, and you can go and make sense out of that. So usually what you talk about by variability when you have a trial structure is you have a difference in mean postural trajectory across trials. Say this is like the reach time series of an arm from multiple trials. You take the mean, you find your residuals around that mean, and then your variance is just the residuals. And we play a little bit of this game, and so I'm not going to talk about this work, but I just wanted to advertise this work from recent paper came out from Tanya Winner and the group, and some more sort of follow-up work by Michael Hess, who are basically thinking about how how human gait dynamics can alter both between able-bodied individuals, between individuals and stroke survivors within the gait. And you can actually do some pretty fancy stuff with that. But that's not kind of the type of variability we're going to talk about today. Particularly, we're going to talk about we have no trial structure. There's no well-defined beginning, no well-defined end. Aligning in time in these types of structures doesn't really work. So how do we make sense out of that? And the general philosophy that we've taken on this is to look for stereotype behaviors. So what do I mean by that? So Hannah said I have a PhD in physics, so let's just put this to use. So imagine this is the space of all postures that an animal can make. Right, this could be, say, like the two joint angles of my PhD was doing insect flight, so I'm used to standing up in front of crowds and doing this a lot. Right. So you can do like some simple motions, you can do some periodic motions, you can do some crazy things. We can do truly crazy things which fill up the whole space. You watch an animal behave, this is not what you see. What you see is something more akin to this. See a few behaviors that are performed often and repeatably with a little bit of noise each time. Some kind of weird stuff every once in a while that you'll see. But for the most part, you see these types of structures. We're going to call these our stereotype behaviors. So stereotype behaviors are behaviors that are performed often and repeatably. And we're going to try and build a representation where, say, each time I go around this ring, I'm going to put a dot there, do this, put a dot there. Then you can watch to see how the animal transitions between all these different stereotype states. That's the idea. So I'm not going to talk about all the methodology, how we do this, because you can see this is quite old at this point, and I'd rather talk about more recent stuff. But the idea is basically we go directly from postural data. You can imagine this is like deep lab cut or sleep tracked limb kinematics. And then we go and we create a nonlinear embedding of that space so that parts of the space where the animal is moving similar parts of its body at similar speeds are plopped together in that representation. So what exactly does that look like? So this is data from roughly 60 male fruit flies. So, and this is a 2D density map in this embedded space. And what you basically can do is pull a bunch of points in time where the animal, say, gets mapped to this little peak down here. And you see that the flies are all running with the same speed. Or you go to this peak up here in the corner, and the flies are all grooming their head. For those of you that can see, it's with a slight left asymmetry. Or down here, the flies are all grooming their left wing. But we call this representation our behavioral space. We have each one of these little peaks is a different one of these stereotype behaviors. I'm happy to geek out about all the details about this later if people are curious. And of course, this is very old. We have some more modern takes on this, and we've done some better things. But this is, for the purposes of today, this is kind of just to give you the picture of what we do here, because this is just, we're going to use this type of representation to then sort of do some interesting stuff with behavior later. And so because of this nature of like, we're mapping this space so that this is where the animal is doing similar things, moving similar parts of its body at similar speeds are mapped together. Like this stuff over here is idle and slow. For example, this little thing up here is proboscis extension, locomotion from fast to slow, wing and abdomen movements, anterior like antenna grooming and things like that. And we can build those types of representations. And just to give you a sense, so these are like all the stuff that we're not going to talk about today, but I wanted to, so one of the things you can do is, this is sort of more like that Heiligenberg example I showed before, where you can see what are kind of the representations of the behavioral repertoire and how are they changing over time through things like circadian rhythm, aging, evolution. This one I almost put in my talk today. So there's a paper we wrote a couple years ago where we sort of really tried to use these methods to reconstruct, to find what behaviors are co-evolving together. It's a really cool story. I'm again happy to talk about that later, or even how you can do behavioral coordination. So you can build these individual maps for different parts of the body, in this case, a rat. So work with Ben Olveski's group, or even looking at social interactions between mothers and infants in a collaboration with Sarah Schultz over at the Marcus Autism Center. I will sort of, just as an example, I'll show a little bit from the agings. This is work from Catherine Overman, the first graduate student in my group that graduated and now has gone from analyzing fruit flies to now she works for the Portland Trail Blazers as a senior data scientist, and now she analyzes the behavior of multimillionaires. Little bit of a change. She won't tell me whose behavior is more complex. And so basically the idea is like we can then go and like by looking to see how these maps are changing, the details don't hugely matter here. But what you can sort of see is that you get different changes between male flies and female flies. We can find low dimensional structures of how those behaviors are changing across age. Long story short, we find that the male flies have a midlife crisis and the females don't. And so this was actually, it was allowed us to make some predictions as to that really its energy budget is the thing which is defining most of the aging patterns in the flies, and that by really focusing in on that, this allowed us to make a bunch of predictions about how energy really is sort of driving a lot of sexual dimorphisms in the flies. And now actually we're working on this in a collaboration with Hong Lu's group, but now moving from flies to worms. That's an example. And this is a neuroscience seminar. I have to show some neurons. Here's some fly neurons. So this was some work we did thinking about this problem. So these are a bunch of descending neurons. So for those of you not used to looking at fly brains, here's the fly brain and here's the fly's ventral nerve cord. And we're looking at descending neurons. So neurons that have their cell bodies up in the brain and have axons that travel down through the ventral nerve cord. And these nice people over at Genelia, like Gwyneth is now at Columbia, but basically, what you can do is optogenetically excite individual neurons through these targeted split Gal4 lines. And because flies' cuticles are translucent to red light, to shine a big honking red LED in the background to excite a neuron and see what happens. You don't have to do any of the crazy surgery stuff that the rodent people in this room have to do. And so what you can do is just sort of cycle this red light on and off at 15-second intervals. And as an example, here's one line. It's not the super cleanest line, but it's going to look cool over here, so I picked this one. So you have a few neurons up here in the brain that travel down through here, and this is starting 5 seconds before the light comes on. Light's going to come on at t = 0, and all of a sudden you see it all goes up there. And at 15 seconds, it's going to go back to normal when light turns off. And so from that, we had to do a bunch of fancy statistics to isolate which of the regions which are upregulated and downregulated, because as you can imagine, it's multiple hypothesis correction hell. And so you basically had to do this, and we can get this really nice upregulated response just in this region. And we get a downregulated response here. This turns out to be idle. So the fly is not doing nothing when you turn on the light. So it was a great positive control. We were very happy. And turns out when you turn on the light, the flies groom their heads uncontrollably. So, and then we're able to go and each time you do this, and so now for each one of these lines, we're able to come up with these different regions, and we're able to go and make sense out of this in terms of trying to get this notion about how does actually behavioral commands get transmitted through this literal information bottleneck, which is the ventral nerve cord, which is only a few hundred neurons.
Gordon,
Yes.
sorry if I missed it. Are these all high-speed video recordings, or do you have labels?
Sorry, do I have what?
For the estimation, do you have labels in?
No, this is all. So for the flies, these are all 100 hertz videos. So I have to recount again. I did insect flight for my PhD, so for me, high speed is 8,000 frames a second. So it's faster than your iPhone usually captures, but it's all based on video imaging. And actually, for the dirty secrets, for the early stuff, this was a lot of these things were done pre the deep learning methodology. And so we actually did a pixel-based approach, which in some ways we still do, because that we still find it can be more robust than actually trying to do the tracking. The disadvantage of that is you lose some degree of interpretability. But I don't know if that answered your question. Yeah, Jeff.
Sorry, this is from a few slides ago, but in your pixel-based approach, how do you handle differences in the quality?
So this is the beautiful thing about flies. Flies are really close to scalar multiples of each other. So for flies, we just normalize by the overall area. And if you plop the flies area on top of each other, it works beautifully well. Now, when we had to do between multiple species, it got a little hairier, and so we had to be a little more clever about it, but it wound up being not as hard as you would think.
That's also true for the males.
Yeah, the males and female, I mean, it's because the body plan, like, there's always going to be some exceptions, but for the most part, the body plan is pretty close to, like is pretty close to being scalar multiples of each other. Minimally, we can take the same individual across multiple. If you chop up that individual's data across multiple chunks of the video and compare that from that individual to other individuals after we do just the normalization by size, I can't tell that as much. So that's the metric I used. Right. Basically, like the thing is just as confused between which one it is. Like if you look at the classifier, it's a chance. So that's how we convinced ourselves we were okay with it. Maybe that just means we did a sloppy job, but but we did an equally sloppy job across everything. So. Great. Good question. Okay. Okay. All right. So that's thinking about how do you think about measuring variability. Really, what I want to focus today is thinking about how do you measure how do you understand variability in behavior across time? That's really the thing I'm particularly curious about. So again, let's go back to this original map. Again, this is just from a bunch of young male flies, and we're gonna see that this applies to other systems, other species. But we're just gonna focus on this because it's where the cleanest example and where I have the prettiest pictures from published papers on. All right, so we're gonna basically take that and I'm a physicist by training, so let's just go and I have a bunch of peaks I'm jumping between. Let's compute transition probabilities, right? So we can go between each of these peaks and compute, given I'm here, where's the next place I go? So this would be the Markov transition probability. So probability, my next state is J, given my current state is I. And here, the thickness of the lines are related to the what that probability is and the handedness. I'm again, physicist, so it's all right-hand rule. Right hand is the direction of transmission. So the idea is what you can see by looking at this is it's not a hairball. Right? You don't see random transitions all across the map. You see mainly kind of local transitions between nearby behaviors. Turns out this is actually for a very boring, dumb reason, which is the fly doesn't want to fall flat on its face. Because if I'm grooming my head, I've got to do a bunch of stuff in between before I start grooming my wings. Otherwise, it's going to be bad. So you basically have these physical constraints which impose this type of structure on the graph. This was actually something that Richard Dawkins noticed back when he was doing all his fly grooming studies. He termed it postural facilitation, and it really sets the structure. And what it allows us to do, one of the common questions I always get when I've given these is, why can you get away with 2D? Why isn't it 3D or 4D or 17D? 2D works because of the fact that you have this planar graph-like structure between, you have to get from one place to another, so you don't wind up getting too many tetrahedrons in this high dimensional space. So you actually wind up having a 2D representation once you do this complicated nonlinear embedding, and it plops out really nicely. I can give you a more technical answer as to why 2D versus 3D, but this is actually sort of why, I think scientifically, why the 2D works. So we have this type of structure, and so now the question is, this is this Markovian picture, right? So I put here, so a bunch of great papers, and actually all these papers I really like, right? So, but sort of the primary thing in modern sort of neuroscience and other sorts of areas where people looking at animal behavior are thinking about Markov models, right? The structure of like, this paper looking at rat grooming, where you see sort of how you go from one part to another. You write this as a transition matrix, or it's work from Jeff's old lab over here, half of Jeff's old lab, thinking about sort of this autoregressive hidden Markov model structure, where you have some sort of hidden Markov model up here, which allows you to move between different types of states, and then that changes kind of how you're moving between sort of postural states down here in the Y. Or you can do some other types of things, looking at like sort of where you combine a GLM with a hidden Markov model to understand how an animal is changing its sensory perception of the world. So, and this is work from Earthy's group. So this notion of underlying Markovianity, kind of one of the key features in how people have thought about modeling behavior. So one of the basic questions is, does this actually make sense, right? Does this actually a good model for how animals move around? So we should actually ask this, as was the idea. So we can ask this question, do behavioral transitions remember? So this is that same data that we plotted on that graph before, as transition matrix, initial state, final state. So this is where you're going, or where you were, where you're going. And basically, well, you can see this nice block diagonal structure, which again sort of shows that modular dynamic that you saw on that previous plot. And so let's ask whether this makes sense. So if you had a Markov model, we take that matrix, multiply it by itself 100 times, right? That's the equivalent of trying to predict 100 time steps into the future. And what you get is you get this thing which is random. Why do I say it's random? Because if you look at the final state, it doesn't depend on the initial state at this point. Right. So you're basically transitioning to a random state, the random probability distribution, and it's not uniform just because you're more likely doing certain behaviors than others. So the point is, after 100 transitions in a Markov model with this particular setup, you should have forgotten almost everything. There's a little bit of structure still up here, but you forget almost everything. But we can actually measure this from the data. We have this joint probability distribution. This is the advantage of having these sort of huge data sets, and you see a lot of structure there. So this is the actual exact same thing. And even measuring 1,000 time steps into the future, you still see lots of structure. It's gotten a little more random, a little more vertical lines, but you still see lots and lots of structure. So we should actually quantify this. I shouldn't just show you pretty pictures and say, trust me. How you can actually do this is you can use something called the Perron-Frobenius theorem and look at the eigenvalues of this transition matrix. The details don't matter a huge amount, but the main idea to remember is that you should have a bunch of eigenvalues which are less than 1, and the larger that eigenvalue is, the longer the effective time scale is the idea. And so what you see for a Markov model, these are all the dashed lines here. And note this is on a log scale on the x-axis. And you can see that the Markov models are dying off much, much more quickly than what the real data is. So here is the blue line and then the equivalent Markov model. And there's a 2 to 3 order of magnitude shift between the two. And in fact, you can measure for longer data sets. And one of my postdoctoral mentors, his lab now has taken data sets where we can measure the fly over essentially weeks. And now you just see that this curve just keeps on moving further out, and this curve stays the same. Yeah, Jeff.
So, I think this is essentially correct, but one thing I've been thinking about is like, what is the right, negative control? Like if I had a robot that obeyed a first order Markov process, but then I filmed it and put it through a large pipeline, how do I know the difference between something that's not a first order model or something where the image
You get occasional errors,
details wrong, and there's some like drifting noise that would generate a slowly decaying curve?
Why would you get drifting if you have where would the drifting, so I could imagine one source of noise would just be misidentification of behavior in that case.
Yeah.
And so that's not going to have a drifting noise, right? Because that's just going to generate perhaps because that drifting noise, if it's just going to happen at any random time, so I should note these lines here are not just multiplying that matrix together 100 times. These lines are dealing with the finite size corrections for the fact that we only have a data of a few.
I see.
So it's actually like you have to do all the finite size corrections and all those sorts of things. So really, like this curve would die here if I didn't have any finite size corrections.
Yeah.
And so this is the much more honest thing. It is theoretically possible that you could, like, if you really had something weird and systematic, like if the light was turning on and off, and if you had a circadian system and like you had the light turning on and off, and that caused a particular error. But this is over an hour, right?
Yeah.
Under kind of uniform conditions.
Yep.
So, and this is also, these lines are averaged across individuals. So
it seems, yeah, definitely.
it seems unlikely that it's just due to, and I didn't include it, but I can show you the individual curves for every individual animal, and this is true for every individual animal we look at. So I agree, like if this was just like some crazy curve that we only saw if we combined a bunch of animals together, we should be really, really worried, but that's actually not what we see.
Cool.
So no, I mean, it's a great question. So this is all stuff we worried intensely about when we were doing this. Yeah, Annabelle.
Mu in this, right?
Oh, mu is just the eigenvalue index. So, and you don't have 1 because it turns out your first eigenvalue just mathematically has to equal 1 because that just says you have a transition probability. That's just the function of it's normalized. So, yeah, Audrey.
So should I be surprised
I mean,
Of course.
a hidden Markov model, you set it up and it can just keep on doing its thing, whereas this fly is in an environment.
Mhm.
It has inputs, it encounters things, maybe it gets hungry.
Long story short, no, you shouldn't be surprised. That being said, it needed saying because if I showed a few slides ago, this is what everybody else does, right? So there's like, and there's also a question about like, okay, am I being annoying? And this is just a problem in the third decimal point that like really we capture most stuff by the hidden Markov model and like we just add a little bit on then because obviously it's not like these things sleep, these things wake up. Like so, but like if you just measure a fly for an hour, like observationally, like if you just put the fly in for the first 10 minutes, it runs around, we ignore that time. Then you start going for the next hour after that. And just by eye, you don't see any changes. Like if you're just watching, it doesn't look weird or different. And you still get all this structure and it's like 2 orders of magnitude bad, right? Yeah. Like if these curves were like here, I probably wouldn't be showing you these plots. I probably would have gone on to something else. And but that sort of is the thing. But long story short, no, we absolutely should not be surprised, but it needed saying. Yeah.
On the same line, then I thought like, for example, this is like ataxia versus Parkinson's. Like some of those have similar characteristics.
Mhm.
Maybe these can be used to discriminate in early onset.
Mm-hmm.
Right.
Yeah, I mean, so certainly there's, I mean, this is, you must have read the first paragraph of my NIH grants. But no, I mean, yeah, so I mean, part of this is thinking about what are the long time scale structure that we see in the behavior, how can this be related to various underlying phenomena that are not directly observable? Parkinson's is a little bit more directly observable because you get things like gait freezing and others, so this might not be the best thing for this, but perhaps early things like mild cognitive decline and other types of issues, you might actually get some interesting things, but yeah. Great. And just for me to show you, this isn't just like this one weird case. Here's flies, male and female flies across all ages. You get that exact same structure. Rats, you get the same thing. This is data from Jesse Marshall and Benza Olveski's group. Or even in human ECOG data. So this is human ECOG data taken over at the Emory Epilepsy Clinic. This is work from a former undergrad in the group. Basically, you get a 5-order or almost a 4-order of magnitude difference between the Markov model and this. If you just put in ECOG data, run our method, and create this little transition map. Again, as Audrey put it very well, we perhaps shouldn't be surprised by all of this, but just to show this is a generic phenomena. But the problem is, and the weird thing is, that this is a super non-generic thing for a model. So one of the things you can, and maybe this partially gets to Jeff's question, is that you could try and write down any kind of normal model, your standard sort of time series model on discrete state transitions you want. Getting 1 or 2 eigenvalues that decay slowly is, you can do this, it's not so hard. Getting lots of them, you have to really fine tune it. You have to really put all those time scales in and be really explicit. You can write a hierarchical hidden Markov model that does all this stuff, but you have to work really, really hard to do that. And so then the question is, what is the sort of model, what's the sort of structure that can actually generate data like that? And that's what we're going to spend the rest of the talk thinking about. And maybe what that means for the neural control of behavior. So let's look at some hints. So let's dig a little bit deeper in what we see. So one of the things we can do is to calculate the mutual information. So again, for those of you who don't know mutual information, it's a nonlinear correlation between 2 different variables. And in this case, I'm going to say, I want to calculate that mutual information between what behavior am I doing now and what behavior am I doing some time step tau into the future. And the data are here in blue, the Markov model is red. The error bars are actually smaller than the lines here because it's average across a bunch of things. I always just like saying that because it's fun. And what you basically see is you get this sort of difference in here, which looks just little, but it's actually, I mean, it's actually quite a big amount in terms of the actual amount of information you have. And what the things that you can see is that how you calculate this is you're actually just doing a sum over a bunch of terms that are related to that transition matrix between what behavior am I doing now, what behavior am I doing sometime tau in the future. And you have this contribution between all those pairwise things. And you can ask, what are the pairs that actually are responsible for this difference? And if you do that, you find that, oh, it's all of these pairs within regions. And so what this implies is these regions are maybe a little sticky. that once you sort of get into one of these regions of space, we tend to stay there longer than a Markov model would suggest, which to my mind has this notion of understanding kind of this immediately brought up of all the Tinbergen I'd read, thinking about hierarchical structure and behavior and thinking about maybe these sort of local interactions might be what's responsible for long time scales. Let's do a little bit of math. So what we can say is we can turn it in, and first thing to do is turn it into a clustering problem. So we can ask what parts of the behavioral space best predict long-term cell dynamics. Let's dig into that. So we're going to cluster the space. So we have a set of states, let's say order 100 states. Now I'm going to cluster them into a smaller number of states. So I'll call that clustering Z of T. Now I'm going to do something called an information bottleneck approach, where you're going to say, I'm going to optimally predict the future. So by getting away all my punchlines. I'm going to optimally predict the future in a way, so by measuring the mutual information between the clustered state and the state in the future, but in a way where I'm going to sort of make that clustering as simple as possible. And then I have this Lagrange multiplier beta, which tells me how much I care about predictability versus simplicity. And you have to do some nonlinear optimization to do this. People have figured this out, it's not so bad. And what you can do is you can do this for different taus. So this axis is how much information, and this is the simplicity. This is how much information. So you sort of want to be up here. This way means simpler, and this way means more predictive. So, and comfortingly, so these are curves from different tau, and as you can see, the curves come down with time. So I can predict less the further out into the future I'm trying to look. I might be worried. I mean, maybe you get some weird periodic signal where you can get some, but for the most part, you should, on average, you would hope these curves are going down. And the question is, what does that clustering look like as we go along one of these curves? So let's pick this tau equals 100 curve that we talked about earlier. And if you go along that curve, what you see as you go from 1 cluster to 2 cluster to 3 cluster to 4 is you get this hierarchical structure where the space, so start with 1, then you break down to doing something, not doing something. Then you have not doing something, or you're doing something or locomotion. Then doing something that's not locomotion is anterior, posterior, and then so on and so on. It keeps breaking down. Or to make the pretty plot that I made exactly one of, because this was a real pain to make, you can then go actually see what that tree looks like as you run through and see as you go from simple to more complex, and you get this hierarchical structure, which is, again, generated from optimally predicting. So what this notion is thinking about the structure of internal states. We're back to Tinbergen again. Thinking and really trying to link together all these ideas, thinking about how this hierarchical structure is actually the thing which is engendering that long time scale structure in the behavior. And so let's go back. So think about this. How might this actually imply? Let's see. How does stereotype behavior evolutionary success? Maybe can you come back to that? We'll ask that again in the question period at the end, because I think that gets to a much deeper set of things that I'll probably ramble on about. So the idea is, let's go back here to these thinking about the behavior of what happens to the behavior when you start exciting these neurons, these descending neurons in the fly. So one thing, so this is a nice clean neuron here. This neuron here goes up here, and you have this symmetric pair. And what we found, and this is true for most of the lines, there's actually multiple regions in the behavioral space which get excited. So in this particular case, we found this yellow region here and this white region here. So, and what's plotted behind it is a conditional density. So this is the density before transitioning to the yellow region. So if I turned on the light and went to the yellow region, what were you doing immediately before I turned on the light? If I turned on the light, you went to the white region, what were you doing immediately before I turned on the light? And what you can see is these are two strikingly different representations. So you have a lot of information. If you tell me what the animal was doing right before, I can tell you which of these two I'm going to go to. And as it turns out, you can actually calculate this. It was 0.5 plus or minus 1 bits, roughly, which, considering in this particular case, the maximum was 0.8 bits. So this is actually quite good. You have a lot of information about which one you're going to possibly go to. This is work from Jiri Shu, a former master's student in the group, I should say. And with the experiments from Jessica Candy and David Stern's group. And so in particular, what we can see is that one particular line, that one white line, if you're doing something with your wings, you tend to do this weird wing extension behavior, which you usually are not going to see in a normal fly just running around in a dish, because this is more related to courtship and aggression, which you're not going to usually see a fly do by themselves. And turns out it's not just that one line. We can go across almost all the lines, and we can see this red. If you're below this red line, that means that you're statistically significant, and we can see that basically about 85% of the lines show this context dependency. And not only that, we later had a theory paper where we talked about how this context dependency can be used to think about optimal transmission, particularly if you have noisy channel in between some large set of behavioral commands and some relatively large number of motor outputs, and you have a information bottleneck connection in between. So this was some other work that I don't have time to talk about, but this is sort of the idea. So the hope is that by thinking about this hierarchical structure and behavior, we could then try and make sense and going back to actually understand what are the types of neural architecture we need to be able to control it and think about it. All right, so for the last bit of my talk, I want to go back to this making sense of variability and behavior across time. And the metaphor that I've had in my head that we've sort of developed, and this is, I should say, this is where we're going to get to the half-baked crazy stuff. So just warning in advance. So you can think about sort of this shifting landscape. Like each of these little basins here are different stereotype behaviors, and then the animal sort of will hop between these different basins. And the overall landscape is then shifting in time as the animal is getting changes in sort of sensory or various internal states, like they're getting hungry, they see a predator, or something, and that landscape winds up shifting around. And so the question then becomes, how do we make inferences about this landscape? How do we make sense out of it? Oh, this.
Interrupt for a second. The stickiness that you saw, is that related to behavioral states, or you think that's completely into behavioral states?
What do you mean? No, it is not. In fact, I'm just going to show you how, and I think it is related. Perfect, great. You've set me up beautifully. So the idea is that, so the first thing you can do is just to show you that we did it, you can try and reproduce stuff with a hidden Markov model. Here's your data. Here's a Markov model. Here's a hidden Markov model. You can get more complicated than that, but you can't really, it's really hard, even if you do all sorts of crazy optimizations to try and get there with it. Like we felt we needed to actually really try and do this well, because otherwise, why sort of keep pushing here? So the idea, what we're thinking is that we wanted to get more dynamical. We wanted to have a less statistical approach to how behavior was changing and thinking more along the lines of dynamical systems, of slowly varying variables of multiple timescales. But the problem is actually fitting that sort of thing didn't make sense initially because we didn't know what to do. So we just did the machine learning approach of, can we throw parameters at the problem? And we did this in like a 2016 sort of way. We trained a recurrent neural network because I'm just a simple-minded physicist and don't really know how to do anything complicated. And so what we did is we basically had this one hot encoded neural network where, say, if you're doing behavior 4, you put in a 1 and all the rest are zeros. You train a recurrent neural network. For those who care, it's LSTM sequence to sequence, 128 units, so nothing super crazy complicated. And then you output some set of variables. You have a softmax layer which samples from those, and then sets what the next state is, and then you try and predict going on. Right. And this was work that was done sort of the end of Catherine's PhD, and then by a postdoc at Ty Pinkovieski. And it's a recurrent neural network. Of course, it's able to reproduce what you would see, right? It's like we just threw millions of parameters, and it was able to figure it out. Awesome. All right. So now, but what we really wanted to do is, because this is a relatively simple model, I mean, LSTMs aren't like, doing analytics isn't going to work, but we can sort of numerically experimentally probe what's going on with this. And I'd also say we get the same information bottleneck clusters more or less than everything. So we're able to create a model which looks like the data. So now let's analyze the model and see what the model has figured out. So one thing we can do is, so we have this structure. Let's just start with one of these things. I'm just going to keep on putting 1, 1, 1, 1, 1, 1, 1 in, and regardless of what the outside state is, and then let the system equilibrate. So we call this our clamped states. So I'm going to go this. So I start from 4, and the system eventually equilibrates down to a particular value. And now we're going to look at what's the internal states inside of the recurrent neural network that it's wound up finding. And if you do that, so this is mapping, so this is a 250, because it's 128 units, each with 2 internal states, for those of you who are LSTM aficionados. We're taking this 256 dimensional thing, use multi-dimensional scaling to get it down to 2D, and these are all of sort of the behavioral states from each of these individual behaviors. If I just clamp it down to that particular state. And these are colored, but they're colored in a way where it's colored by which part of the behavioral map did it come from. And as you can see, it's basically more or less reproduced the behavioral map, which is actually impressive because we told it nothing about the spatial structure of the map. We just said it's behavior 1, it's behavior 17, it's behavior 35. And so the high dimensional structure of the internal states of the RNN have rediscovered this behavioral map structure. So that tells us the behavioral map is actually telling us something deep about the underlying dynamical structure for trying to predict sequences in the map. Jeff, go ahead. Yeah, Jeff.
Yeah, just to make sure I'm following. So here you're just predicting the statistics of what comes next through the LSTM. And
well, it's the sequence. It's really it's sequence to sequence, or predicting like the statistics of like multiple time steps into the future.
how many time steps into the future?
I think in this one, it's each sequence is 1,000. So you're trying to predict what with some amount of lag. So I think it was like 1 to 1,000 and then 11 to 1,000 and 11 to 1,010 or something.
Gotcha. And I guess what I'm trying to understand is, so you have stickiness, and then occasionally you hop out of idle into slower.
I haven't talked about stickiness yet, right?
Well,
In this.
yeah, assuming there's some stickiness. Yeah, could you get a picture that doesn't look like this? You know, assuming that you have a couple of those features from the previous transition.
Probably not.
Yeah.
But again, the point is we're going to do something we're going to actually impose that directly in a minute,
Cool.
right? I'm showing this just to inspire kind of just doing a bit of math at the end.
Yeah.
Right. Yeah, but I mean, this is exactly how we were thinking about things, but we wanted to just make sure that we actually were thinking about the problem properly, right? But I mean, it's a great question. And so we have all this structure. And so again, this is very reminiscent and thinking about the sort of transition structure and thinking, as Jeff said, the stickiness that is inevitable. And so we're reproducing this, just not without telling it any of that. It's able to reproduce that stuff. So we have a reasonable model, and that model uses that information in the space that we were seeing. So the second thing we can do is now, so when our original model, we had the softmax layer, which was injecting some amount of noise into the system. Now we can make this a Langevin model and just add a little bit of Gaussian noise and feed that back. So now if we're going to start from one of those sort of states that I said, you can add a little bit of noise, run it around a bunch of times. And what happens if you do that is you actually get this nice kind of, you get these nice Arrhenius law type dynamics, where you can actually see I'm in one of these states, and if I look at, I should see that my persistence time within one of these states should be related to some temperature times energy barrier. So I can go and fit a slope to my persistence times as I change the temperature in this model, and that temperature is just the amount of noise I'm adding in, and I get this, and then across all states, I get this type of slope. So we get this model of, I have this basin hopping type model. This is no longer sort of this absurd idea, but this isn't quite right. Because we know, for those of you, again, who might be aficionados of this sort of thing, if I have a bunch of basins and I have some random sort of noise injecting and probability of transitioning to the next state, this is a Markov process. So the question then becomes, can we infer not just how we're moving from one state to another, but how is this thing warping and moving with time? Right, so really focusing on this part, sort of how does this underlying landscape change? And so now here comes the truly half-baked parts of things where we think about how maybe to do that. And so what we did is, thinking about this, we said, let's actually just measure something. So what we did is, let's actually estimate what that transition, how the transition probabilities are changing with time. It turns out this is a little more calculated than just taking a sliding window and calculating it, because if you don't see a given state and area, you have much better estimates for a certain part of your transition matrix than others who have to figure out a nice way of imposing a prior, and we have a way that at least we're happy with, but maybe there's some better ways to do it. But the long story short is we have now a transition matrix as a function of time. We could just measure that directly from the data. There's no RNN, no nothing. This is just back to the data. And if you now plug how that transition matrix is moving as a function of time, and then simulate data based on that time and varying transition matrix, you now see kind of how you can now reproduce those types of eigenvalue decay structures that we saw in the real data. So by having this time varying transition matrix, again, we perhaps shouldn't be surprised, but this particular form of time varying transition matrix reproduces this structure. So that leads to the question, how is that transition matrix changing in time and what's the structure? So what we did is we then sort of took inspiration from what we just saw, is we can then do a little bit of math. It's actually not too bad. It's just a geometric series of going and calculating basically what we think the energy barrier is as a function of time based off of the various transition matrix elements. And it actually turns out to be this relatively simple function over here. And we did some other calculations to show that we can just basically assume that the temperature is constant. So we're going to be basically just focusing to see how do these transition matrix elements change with time. And that changes to basically these energy barrier heights, or how big are these barriers between the little basins. And first basic thing we did is we just do a PCA of how those delta EIJs are changing. And immediately you see this modular structure pop right back up that we saw earlier when we were looking at the transition matrix. And that we saw from just looking at the raw behavioral data. And so this tells us maybe we should go and look into the modular structure that we're seeing. Just a couple more slides. And so what we did is now let's actually go and let's coarse grain. This is one of the things you need. I think that's going to be one of the key features in any of these types of behavioral models is this ability to coarse grain between finer and coarser scales. Let's coarse grain up just as idle and slow versus active. You're doing something. And looking at how do you move between those two sort of coarser states. And we can calculate that, and this is just data from one animal. And you can see these large scale fluctuations between those energy barriers, function of time. And note, really that energy barrier, the transition probability goes like the log of this. So these changes of like 2 are orders of magnitude changes in the transition probability. So you're getting lots and lots of fluctuations. So for example, for here, you have a relatively low barrier from going from active to idle. So you're getting sleepier. And here you're getting more awake because you're really trying to push. Once you're in the active state, it's getting really, really hard to be in the idle state. Or over here, you can go at a slightly less coarse. You now have this 3 state, and now you can really start to see how the structure, like comfortingly, you can see this other to idle. You can see sort of how a lot of these going from, if you were to, I didn't plot it here, but if you were to plot the barriers between idle and locomotion, they're like way up here. It's like you never see any transitions between those two. Oops. All right, so you never see any transitions between those two, and you can sort of see how you get these fluctuations at different scales. And just to sort of show, this is what happens as a function of time, as you actually try and estimate what those barriers look like. So here is idle, other, and locomotion, and time, or I think this is a little easier to see. Here's just a movie of how it fluctuates. Again, this is just for one fly. So you can just sort of see how these barriers shift around and move, and that essentially these delta E's, and my kind of claim is that this can be then used as an internal state measurement that we can then use to then correlate against various sort of changes in neural activity or gene expression activity or whatever internal state you feel like measuring that has time courses along these types of scales. And so it's a way of going directly from the data of having what are the internal state structures that actually are regulating of larger scale shifts in behavioral function. And that's sort of the goal. And so one sort of very simple thing to get back to the stickiness idea is we should actually confirm this, right? So we'd expect if really we're seeing the stickiness, the energy barrier should increase with time as sort of the longer you stay in the basin, sort of this memory foam like idea. If you're there, the basin is going to decrease. And sure enough, you actually see this. So I'm just showing this on the coarsest level. You can show it, see it in other ones. So this is a 2D histogram of this is your delta E idle to active. And this is what you were doing basically given that what's your probability of basically how long you've been in the idle state. So the longer you've been in the idle state, these energy barriers wind up increasing. And the same is also true. The longer you've been in the active state, these energy barriers on average increase quite a bit amount as well. And so the stickiness seems to be this key feature. And then eventually, either through some dynamical process or through flipping back across randomly, you wind up getting this. And so where we're sort of, and this is where I'll more or less end, is thinking about, like, that's really where we are right now on this idea. Is how do you actually go and model those fluctuations in the landscape? We can now have this way of measuring it through this basin level fluctuation, but really the question is, what does the model of that look like? How do we actually understand how they're changing with time? Does it just response from external inputs, or is there really some internal driving and so this is, or seeing how external inputs then affects and interact with these sort of more native internal states that we're seeing. And with that, I would like to thank all the great people in the group. Particularly, I showed work from Michael, Taniel, Kanishk, Jiri, Catherine, and Itai today, and all the people that have given us money. So thank you. Simon.
I think right at the end you were getting to it, but the attributing the stickiness to its internal state is very evocative and sort of what we really want to do as neuroscientists. What about with the environmental interactions, especially in changing environments? Because it seems like that could also provide that context. And it seems then like if we have this sort of measure that's predicting sort of the longer timescales fluctuations, it might be dangerous to attribute those to internal state, but what really could be changing is the other state, the interaction of the body with the environment. And you know, there's a long history of forgetting about that.
Well, no, I should note, these are a bunch of, as my colleague David Stern likes to call it, the bored fly assay. In these particular cases, it's not to say they're not interacting with their external environment, but it's a fly crawling around in a dish.
And the environment is pretty stationary.
Exactly.
Whereas if we took natural behavior, it wouldn't be. So that's what I'm concerned about, is how much of if we can extend this to a regime where, but even in there, the mechanical interaction with the environment is changing.
Right.
Because the transition from one behavior to another may depend on the biomechanical flows, for example, right? And that would change.
Well, yeah,
Static.
I mean, the obvious flaw in all this stuff is the animals aren't doing the one that's explicitly engineered to do the one thing that the animals are named after, which is fly. Right? So we're obviously, and you probably are going to get a more complicated structure once you have these additional context and other things. But this is what sort of maybe what allows it to be two-dimensional and allows us to do a bunch of simpler things. But
do you think you can attribute, the key thing is, do you think you can attribute the energy fluctuations to the internal state in that context? Because I would have the challenge of taking that into my experiment and applying that technique is if I wanted to attribute the fluctuations to internal state fluctuations, but if an alternative source of those fluctuations is potentially environmental interaction, it'd be nice to separate those. I wonder if we could.
I mean, there's two things. So I would say, so there's parts of it which are, there's different types of environmental interactions. Like, I agree. I honestly don't know how to deal with the changes in mechanical interactions with the world, like, which is obviously a huge issue. But I think if you think about your external interactions, be say like social behavior or something like that, then there might be possibilities of understanding by actually understanding like how information is flowing from one animal to a different animal, or how the structure is going. And so this is actually a project from a couple of different people in my group who are thinking about this on sort of applying these types of things for understanding how changes in social behavior are related to changes in internal state. But I think at the end of the day, really a lot of it is just doing a bunch of controls. And
people are thinking about the flow of information from an environment, for example, too.
Well, yeah, no, I mean, many people are, but like in this particular framework, I don't necessarily know how to do that. Right. That's not to say there's not a way, but I haven't thought of it. Yeah, Anna.
So I was just wondering how you make sure that you are on the right time scale when you're looking into the eigenvalues of the transition matrix. How often you update, and how do you make the comparison of the data of the transition matrix from the data versus the Markov? So like that should depend on the shape. The actual shape of the decay should depend on the actual time scale delta t, right?
Well, you pick for different. Oh, that's a great question. So what you basically what you're doing.
How do you make sure that it's fair comparison?
Yeah, I mean, so what you're actually doing. I have a lot of slides. So what you're actually doing here is you basically each of these is a pairwise comparison. So you're not actually doing this across time. You're just saying here to here, 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 here to here. So you're just always computing a joint probability distribution each time. So that's why it kind of works. And so yeah, because this isn't like a predictive information calculation. We're actually trying to get like how much information are you doing across the whole past of the system, right? Yeah.
Thanks for first. For the linear state, you consider, if you categorize it as a deterministic and it's stochastic. Have you considered chaotic behavior as well in your course?
I mean, chaotic, it's sort of, so you can think, I would say the closest you can think about in chaotic behavior is you probably can do a decent amount of these types of models through some sort of reservoir computer that has like underlying chaotic structure. But if you look at the behavior, I think the stochastic component is high enough that thinking about it as a chaotic system is tough. Now, my colleague, Greg Stevens, has actually done some beautiful work showing how chaotic behavior, how you can use Tockin's embedding theorem. And we've done some of this on other projects, but how you can use Tockin's embedding theorem to understand C. elegans behavior. But that's sort of, in some ways, a very special case for, I think, specifically because of the biomechanics of the problem, because you're effectively at a low Reynolds number. And so you don't have a lot of, they're way more constrained by posture than, say, a fly is in a lot of ways. So I think and they're going directly trying to model postural time series, which is not what we're trying to do here. We're throwing away the posture and going to this other representation where we're looking at position in this overall space. So chaos, at least in my mind, isn't quite the right tool to look at this mathematically, but there is sort of this multi-timescale structure that chaos also has with a bunch of unstable fixed points and things is, I think, spiritually similar, but mathematically tricky to use to actually perform these types of models. Okay, great.
So, how does the stereotypical behavior relate to the success of, say, evolutionary success?
I mean, clearly it does, right? Because otherwise, these stereotype behaviors have evolved and exist because they're things that perform some function in the animal's life. Locomotion, for example, is a great example, or flight and the fly. So obviously, many of these particular stereotype behaviors have specific evolutionary functions. The question then becomes, what's the evolutionary function of how these stereotype behaviors are tiled together? And that's really, I think, one of the things that we're thinking about. We had this paper where we were trying to find which behaviors were co-evolving together, and we have some methodology for doing that across species, and then really thinking about those types of structures. And that's really the thing what I think might be, or at least where my group is thinking more about.
How does the transition between behaviors inform about the neuronal circuits involved in control of the behaviors or transition between the behaviors?
I hope so. I hope so. I mean, this is one of the things we're trying to do. Like, as I showed for this one example from the fly descending circuitry, right? There's the circuitry in some places seems to be set up so that you have this context dependence because of the fact that you have a limited bandwidth structure. And so for example, what I didn't show is like the work from Gwyneth Card's lab, where they showed that the same neuron has very different functions and when the fly is walking or versus it's flying, and how basically the system completely multiplexes itself and sort of allows it through sort of local connections, local interneuron circuitry that allows it to go and actually do these sort of complicated structures. So yeah, we think about a lot of these things, but again, we're in sort of early days.
Thanks, Gordon.
Thank you.